Hello, welcome to my presentation. My name is M.Moura, and I work at Por Lab at the University of Oslo in Norway. And I'll be talking about thin film flow, fluid transport via thin liquid films in slow porous media flows. This is a work that I have done with my collaborators here, Knut Jørgen Molloy, Eirik Flekoy, Gerhard Scheffer, and Hanno Toussaint from the University of Oslo and the University of Strasbourg. Okay, so let me see first if this works. If I manage, you see that there is a TV there. Let's see if I manage to turn it on like this. Maybe, okay, is it working? I guess it's working, huh? Right, so we go like this. Right, okay. So it's very convenient for me that we have this background at Por Lab because it's actually the start of the kind of experiments that I work with. So it's very easy for me to make the presentations. So you see these kind of things here. These are drainage experiments. So air is entering from the top and going down like this, and it leaves the system on the bottom there. And what happens here is that there are three different draining speeds. So it goes from fastest on the left to slowest on the right. And this is a work that has been done by Grunde, Lovo, and others in 2004, the one in this image here. And in the experiments, if we do it, it looks a little bit like this. So you see the porous medium there is this glass beads here, and then air is starting to invade. So that's how it looks like. Just let it run a little bit for it to get a little feeling for it. The liquid there is a mixture of glycerol and water. So it's a more viscous fluid than the invading air, and it's also the wetting phase. So those are drainage experiments. So as I said, this goes from fast on the left to slow on the right. And one interesting thing to see is that whenever you have this slow experiments, you typically have these trapped clusters. So those are commonly found in this slow drainage experiments. You can see how it goes, the trapping, how it happens like this in this video here. So this is 10 times faster than the real life, and the model is tilted by 20 degrees like this. And you see that whenever the air phase surrounds the liquid phase, the black phase there, it creates a trapped cluster like this one here. And they're very, very frequent, very common. There's another one there. And the important thing to see is that this trapping, typically when it happens, it isolates the fluid from the main drainage cluster, this one here, the defending cluster, and then that fluid is basically disconnected from the outlet. So it doesn't go anywhere, it just remains trapped there. And this is something that people kind of knew for quite some time, and it's incorporated in the numerical models used to simulate these things, like the invasion percolation algorithm is one of the numerical models, and it includes a trapping rule for like that. So whenever the fluid is trapped, so invasion doesn't happen anymore on that cluster. But if we look close enough and for long enough, sometimes we see things like this happening. So this is exactly the opposite of what I just said. This cluster here, you see, it's been trapped, but for some reason it got drained. And this is the kind of invasion event that I'll be talking about in this presentation. So it's different from the regular invasion event because drainage here is not happening through the usual pathways, the usual path of pore throats and pore bodies, because all of the pore throats around here, and pore bodies, they are filled with air, they are filled with the other phase. So something else is happening there. There's some other mechanism to explain this drainage. This is what we're going to study. So the experimental setup is like this. We use a glass bead model like that, and it's a monolayer of glass beads. The beads are about 1 millimeter in diameter. This is a modified helical cell. It has a fixed top plate like that. It's a glass plate. And on the bottom plate, we have a cushion. It's an air cushion. And the purpose of this cushion is to press the beads against the top plate so that they don't move. And then, because there is a small experimental variation on the size of the beads, you always have that. And then if there was a regular cell with the two fixed plates, there could be some beads moving around a little bit. So with this modified cell, we circumvent that problem. And then we withdraw the liquid like that with a syringe pump here. And the inlet, the other side of the model, is open to the atmosphere. So pressure in the drain, in the non-wetting phase, in the air phase, is always atmospheric pressure. Air pressure is always atmospheric pressure. And then we are withdrawing the liquid. So this is a zoomed in view of the system. Okay, and we take pictures from the top like that. Right, so in these systems we can have imbibition, that's when the wetting phase invades the non-wetting phase like that, and drainage, that's the opposite, when the non-wetting phase invades the wetting phase. We'll be focusing on drainage. The invasion mechanisms are very different from one to the other. You see here you tend to have more compact invasion, don't really form that many isolated clusters. And here you have this invasion in the sequence of bursts. So this is kind of a more continuous, this is an intermittent process. And that's the one that we're going to study here. Okay, so we can come up with this very general and somewhat useless sentence here that transport happens along a network of pore bodies and pore throats. I mean, this is pretty general. We know that very well in porous media science. And this network, I mean, there are several ways to extract it and to define it. So this is, I'm just zooming in on that square there. And one way to extract the network of pore throats and pore bodies here is to show this Delaunay triangulation. So we just get the glass bead centers like that, it's the blue dots, and then we produce the Delaunay triangulation. And it's this red lines here. And the network is actually the dual graph to that Delaunay triangulation. It's called the Voronoi diagram. So it's the green lines here. So this is one way to get the network. Okay, and I mentioned that this was not being drained by that usual network. So what's happening here? What kind of mechanism is behind this drainage? And then if we zoom in well enough, you see that there is some very thin films of liquid that are still connecting that body there to the defending phase. So if this square here, I'm zooming in around here, that's just below the cluster there. And then we see that in between the beads, you have these tiny bits of liquid, and those are capillary bridges. Capillary bridge, so this is a zoomed in version here. You see there is a little bit of liquid there, and a capillary bridge, you can make one if you just put your finger on water like that, and you lift. So you see that there's a little bit of a liquid that remains there. This is the capillary bridge. And we believe that this bridge, they are acting as paths for the drainage of that cluster. So this is one of the mechanisms that can explain why this cluster is being drained. Of course, it doesn't necessarily need to be the only one. You can also have other things that we don't see. For instance, there could be a very thin film of liquid in some places around on the plate itself. So that would be another mechanism. But the point here is there is more. There are other mechanisms that can lead to drainage other than the usual transport through the regular network of pore bodies and pore throats. So that sentence there, that transport happens along the network of pore bodies and pore throats, that changed to it happens mostly along a primary network of pore bodies and pore throats. But a smaller part of the transport also happens along a secondary network of capillary bridges and thin liquid films. That's the kind of things that we study here. So we can identify the drainage events, the pore invasion events that happen due to film flow. And that's how we do it here. You see, this is a snapshot at some instant here. So what's white is the air phase, and in black is the porous medium and the liquid as well. And then we can get the front like that, and you see that most, so in light blue here, this color there, it's what happens between one image and the following. So you see that most of the invasion happens along the interface here, ahead of the interface. So this is the regular invasion. But you see these invasion events here that happen behind, inside the air cluster. So these are the film flow invasion events. And if we track just those, it's the yellow ones that you see here. You see that it's not so infrequent. I mean, it happens quite a lot of them. And so in green, you see the regular invasion events, and then in yellow is the film flow invasion events. And in this case here, it was about 8.3% of the total saturation increase. So this is not negligible. This is something that we have to consider. It is a lot. This is an invasion time map. So since we have access to the whole dynamics, we can just plot the time of invasion of a given pore. And then you have this nice rainbow plot here, and you see that the front moves more or less at a constant speed. That's the gradient of the rainbow, but you have this isolated invasion events with different colors there, different from the surroundings. So this invasion here happens much later than the main front passed by this point here. So the main front passed around 70 hours there, and the invasion is around 20 hours. So this is a film flow invasion. And we can make a histogram of the sizes of the capillary bridges and of the pore throat sizes. And that's what we have here. So in the yellow is all the pore throats, and in blue is just the size of the bridges like this. So you see that the bridges tend to stay at the smallest pore throats. And this is something that we would expect, right? If the pore throat is too wide, then the bridge turns unstable and it cannot stay there. And this instability when the throat becomes too wide is connected to the Rayleigh plateau hydrodynamic instability. That's the instability on the tap. If you open the tap, then you see this stream of water, and then after it gets long enough, it starts to break down into droplets. So I think this is what's happening there too. If the bridge is too long, it's not stable, and then it breaks into a film coating one of the beads and a film coating another of the beads. And we can estimate the maximum size of these bridges if we use the scale of the experiment. And this is the radius of the bridge there. It's about 15 millimeters if we just measure from the image. I mean, it varies, but it's roughly around that. And we can estimate, using the condition for the stability of the Rayleigh plateau instability here, that the diameter of the bridges would be around half a millimeter at the maximum diameter. And that's more or less what we see here. Like for more than half a millimeter, we don't really see bridges. So this is more or less consistent with what we're measuring. We can also measure the condition probability of bridge formation, and that's just the ratio of these two histograms. So this is just measuring that if you have a throat of a given size, what's the probability that it will hold a bridge? So it's like that. The details of all of these things, the calculation and so on, I'm not given here, so you can find it on the paper. It's this one here. Right. So one of the main findings of this paper is that there is an active zone which follows the main drainage zone, and that's where most of the film flow invasion happens. So it's like that. This is a video here, and whatever you see in red is a film flow drainage event. And you see that it tends to follow the motion of the main front. And here we just have the position of the main front in green and the position of the active zone in black, and you see that one is following the other. This subplot here is the widths of these two zones, the width of the interface and the width of the active zone, width measured in this direction, in the vertical direction here. So we have found that we can measure the probability of film flow activity in the reference frame moving with the front, and we found it to be like this. So this is for experiments of varying angles, and in all of them, the film flow activity happens closer to the front, and the steeper the experiment, the more well-defined that is. I mean, let me say that the width of this zone here, where you have film flow events, it gets reduced and it gets more closer to the main front. So we have found also that if you consider an analysis inside subwindows like this, so this is a subwindow at a fixed Y position here, the graph of the air saturation as a function of the distance to the inlet, so as a function of Y, is like that for different times. And if we just consider a vertical slice here, so just this part here, so that means to say it's a constant Y there, so it's a constant position of the subwindow. Then if we wait long enough, you go from this green to the black there, so the green one is at 18 hours, and the black one is 77 hours, and that means that the saturation in that subwindow is increasing. So this increment is due to film flow. So another thing is that we can now track this capillary bridge network, this secondary network. So we have devised a machine learning algorithm to do that, and we're basically trying to track one pattern of color intensity along a line perpendicular to the bridge like this. So we get to the beads. So for all beads, we consider this thick line here perpendicular to them, and then we see the color intensity along that line. So if it's air filled, then you have a high intensity. If it's liquid filled, you have low intensity. And if it goes like a capillary bridge, you have something like that, from white to black to white, high intensity, low intensity, high intensity. So it's this kind of pattern that we're trying to track. And we use machine learning to do that, and this is the kind of things that we obtain now. So the blue dots here is just the center of some of the beads, and the green dots is the center of some of the clusters there, and you see the whole connectivity of the system this way. So the challenge is to do this for the whole network, of course, and this is convenient because it can show you what are the clusters that are connected to the main drainage cluster. So here you see that this cluster C1 is connected to C0, so it can be drained through this capillary bridge there, and C2 is indirectly connected to C0. It's connected to C1, there is connected to C0 like this. So what we want to do is to do this, of course, zooming out for the whole network. And if I zoom out enough, then you don't understand anything because it's too big. It's like this. And once we do that, we can map what are the clusters that are connected to the main invasion body like this. This analysis is still a little bit problematic because if you just miscount a few of the bridges, then the connectivity gets completely messed up. Right, like if this bridge here was not identified as a bridge, then this whole cluster would be lost. I mean, we would not see the connection of this whole cluster with that one. And then since this one is indirectly connected to that one, the connection of that one would also be lost. So it's we're still fiddling a little bit with this analysis to make it work, but we can have an idea more or less of how it's going to look like. We see that it's mostly the clusters close to the main drainage cluster that are connected to it, and those are the ones that are drained due to film flow. So that's consistent with what we have found, that there is this active zone close to the main drainage zone. And then once we do that, it's pretty convenient because we can use graph theory. We can apply arguments of graph theory to this problem. And we can use, for instance, this is a directed graph showing there are some arrows here, and then you can show the direction of the flow that connects any of these pores and these isolated truss clusters via the film flow network through the main drainage body. So our conclusions is this: we have experimentally measured imaged the film flow drainage events. The majority of these events are concentrated along an active zone that follows the motion of the main interface, the liquid-air interface. And we have devised a preliminary machine learning algorithm to track this film flow network. So the main reference for this work is this paper here, and I would like also to point out to this other paper where we have analyzed this intermittent dynamics of these drainage experiments a little bit more. Okay, so to finish, I just want to advertise this is the Interpore Norway Gallery of Porous Media Art. You see lots of these images and experiments are quite interesting, and even if you don't understand the physics and the science behind it, porous media science has a lot of very nice things to see just for the aesthetics of it. So we have a gallery, an online gallery, and if you want to contribute to it, we're very welcome. Okay, so that's it. So thank you very much.